It's Handsome ASMR, back with another video. This time, I'm going to be explaining the difference between gram-positive and gram-negative bacteria. So I'm going to provide an overview of that. So, one of the most important differences pertains to gram staining reaction. For gram-positive bacteria, they will retain the crystal violet stain, and so this means they will appear purple under the microscope. Gram-negative bacteria, on the other hand, do not retain the crystal violet. Instead, they will take up the saffron and counterstain, which means they will appear pink or red on the staining. So that's one difference. Another important difference pertains to the cell wall structure. So for gram-positive bacteria, they consist of, or its cell wall consists of, thick peptidoglycan layer. And this will contain what is called teichoic acids. These happen to be structural and immunogenic. There's also no outer membrane. And then for gram-negative bacteria, they will actually have a thin peptidoglycan layer. And this particular cell wall is going to be surrounded by an outer membrane containing the lipopolysaccharide, or LPS. And that is, once again, the outer membrane, as we can see over here. So therefore, there will be a periplasmic space between the inner and the outer membrane. Another important difference pertains to the outer membrane and the lipopolysaccharide presence. Once again, gram-positive has no outer membrane or lipopolysaccharide. Gram-negative, on the other hand, has an outer membrane, and it will also have the lipopolysaccharide, which can basically be an endotoxin. So this is important for constituting a virulence factor, which means it can trigger a septic shock. Some other difference pertains to antibiotic susceptibility. For gram-positive bacteria, they are generally more susceptible to beta-lactams, such as penicillin, and that is because of the exposed peptidoglycan cell wall. So, treatments for gram-positive bacterial infection includes beta-lactam medications like penicillin. Gram-negative bacteria, on the other hand, are more resistant due to the presence of that outer membrane barrier we talked about earlier. And so, this may require broad-spectrum antibiotics for treatments like cephalosporins or carbapenems. Finally, we can talk about some examples of gram-positive and gram-negative organisms or bacteria. Gram-positive bacteria include Staphylococcus, Streptococcus, Clostridium, Listeria, and Bacillus. And then for gram-negative bacteria, they can include Escherichia coli, Neisseria, Pseudomonas, Salmonella, and Klebsiella. And then for some clinical stuff to think about, gram-negative bacteria are more often associated with sepsis due to the presence of that lipopolysaccharide. Gram-positive bacteria, on the other hand, will more commonly cause skin and soft tissue infections, respiratory infections, and toxic shock. And this is especially the case with the Staphylococcus aureus. So, to summarize what we talked about, there's some ways to distinguish between gram-positive and gram-negative bacteria. Gram-positive bacteria will appear purple on a gram stain, while gram-negative will appear pink or red on a gram stain. Gram-positive will have so that means the gram-negative will have the lipopolysaccharide, which can be an endotoxin, while the gram-positive won't have one. On the other hand, the gram-positive bacteria will have teichoic acids present on its cell wall, while the gram-negative won't have one. And so, clinically speaking, it's important to remember that gram-positive bacteria are more sensitive to antibiotics, while gram-negative tends to be more resistant.